Hi dear students, today let's see the checkpoints and regulation of cell cycle. Regulation of the cell cycle involves processes crucial to the survival of a cell and that include the detection and repair of genetic damage, the prevention of uncontrolled cell division. There are different regulators for this purpose and that includes the cell cycle checkpoints, cyclins and CDKs, certain inhibitors and the E2F and RB proteins. Checkpoints are surveillance mechanisms that halt the progress of the cell cycle if any of the chromosomal DNA is damaged or certain critical processes such as DNA replication during S phase or chromosome alignment during M phase have not been properly completed. Checkpoints ensure that each of the various events of the cell cycle occurs accurately and in the proper order. Checkpoints are activated throughout the cell cycle by a system of sensors that recognize DNA damage or cellular abnormalities. If a sensor detects the presence of a damage, it triggers a response that temporarily arrests further cell cycle progress. The cell can then use the delay to repair the damage or correct the defect rather than continuing to the next stage. If the DNA is damaged beyond repair, the checkpoint mechanism can transmit a signal that leads either to death of the cell or its conversion to a state of permanent cell cycle arrest known as senescence. There are three main checkpoints that control the cell cycle in eukaryotic cells. They are the G1 checkpoint or G1 restriction point, G2 checkpoint and the metaphase checkpoint. Let's see what's happening in G1 checkpoint. G1 checkpoint is the main decision point for a cell, that is the primary point at which it must choose whether or not to divide. Once the cell passes the G1 checkpoint and enters S phase, it becomes irreversibly committed to division. That is, barring unexpected problems such as DNA damage or replication errors, a cell that passes the G1 will continue the rest of the way through the cell cycle and produce two daughter cells. At the G1 checkpoint, a cell checks whether internal and external conditions are right for division. And there are some factors a cell might assess. The first one is the size. It checks whether the cell is large enough to divide. And then it checks the nutrients. That is, does the cell have enough energy reserves or available nutrients to divide? It also checks for some molecular signals, like if the cell is receiving any positive cues such as the growth factors from neighbors. Then the DNA integrity, whether any of the DNA is damaged. If a cell doesn't get the go-ahead cues it needs at the G1 checkpoint, it may leave the cell cycle and enter a resting state called G0. Some cells stay permanently in G0 while others resume dividing if conditions improve. Next is the G2 checkpoint. It makes sure that the cell division goes smoothly. That is, it produces healthy daughter cells with complete and undamaged DNA. At G2 checkpoint, it will check about the DNA's integrity, if there is any DNA damage or what. Then the DNA replication, that is, was the DNA completely copied during the S phase. If errors or damage are detected, the cell will pause at the G2 checkpoint to allow for repairs. If the checkpoint mechanisms detect problems with the DNA, the cell cycle is halted and the cell attempts to either complete DNA replication or repair the damaged DNA. If the damage is irreparable, the cell may undergo apoptosis or programmed cell death. Their self-destruction mechanism ensures that damaged DNA is not passed on to daughter cells and is important in preventing cancer. Next is the spindle checkpoint or the M checkpoint. Here the cell examines whether all the sister chromatids are correctly attached to the spindle microtubules. Because the separation of the sister chromatids during anaphase is an irreversible step. The cycle will not proceed until all the chromosomes are firmly attached to at least two spindle fibers from opposite poles of the cell. It seems that cells don't actually scan the metaphase plate to confirm that all the chromosomes are there. Instead, they look for triangular chromosomes that are in the wrong place. Example, that is floating around in the cytoplasm. If a chromosome is misplaced, the cell will pause mitosis, allowing time for the spindle to capture the stray chromosome or the floating ones in the cytoplasm. Cell cycle regulators are broadly classified into two types. The positive regulators, they are the ones that control the changes necessary for cell division, and the negative regulators are the ones that control the positive regulators. Let's see about the positive regulators of the cell cycle. The cycle starts when a growth factor acts on a quiescent cell, provoking it to divide. Quiescent cell refers to the inactive or dormant cell. Then the growth factors stimulate production of the cell cycle regulators, which are coded for by the delayed response genes. There are two families of proteins, cyclins and cyclin-dependent kinases, that is CDKs, which control the progress through the cycle. The CDKs functioning sequentially phosphorylate the various proteins, for example enzymes, activating some and inhibiting others to coordinate their activities. Cyclins are among the most important core cell cycle regulators. Cyclins are a group of related proteins, and there are four basic types found in humans and most other eukaryotes. That is G1 cyclins, G1S cyclins, S cyclins and M cyclins. As the name suggests, each cyclin is associated with a particular phase, transition or set of phases in the cell cycle and helps drive the events of that phase or period. For instance, M cyclin promotes the events of M phase, such as nuclear envelope breakdown and chromosome condensation. Cyclin-dependent kinases. In order to drive the cell cycle forward, a cyclin must activate or inactivate many target proteins inside of the cell. Cyclins drive the events of the cell cycle by partnering with a family of enzymes called the cyclin-dependent kinases. CDK is inactive when it is alone, but the binding of a cyclin activates it, making it a functional enzyme and allowing it to modify target proteins. CDKs are kinases, the enzymes that phosphorylate specific target proteins. The attached phosphate group acts like a switch, making the target protein more or less active. When a cyclin attaches to a CDK, it has two important effects. The first one is it activates the CDK as a kinase, but it also directs the CDK to a specific set of target proteins, ones appropriate to the cell cycle period, controlled by the cyclin. For instance, G1S cyclin sends CDKs to S phase targets, for example for promoting DNA replication, while M cyclin sends CDKs to M phase targets, for example for making the nuclear membrane breakdown. In this image, you can see that in the absence of cyclin, the CDK is inactive and it cannot modify the target protein, so the S phase factors remains off. But the binding of cyclin activates it and making it as a functional enzyme and allowing it to modify the target proteins, that is why it is phosphorylated, and the S phase begins and the DNA replication enzymes are activated. In general, CDK level remains relatively constant across the cell cycle, but CDK activity and target proteins change as levels of the various cyclin rise and fall. In addition to needing a cyclin partner, CDKs must also be phosphorylated on a particular site. in order to be active, and may also be negatively regulated by phosphorylation of other sites. A famous example of how cyclins and CDKs work together to control cell cycle transition is that of maturation promoting factor or MPF. Like a typical cyclin, M-cyclin stays at low levels for much of the cell cycle, but builds up as the cell approaches the G to M transition. As M-cyclin accumulates, it binds to CDKs already present in the cell, forming complexes that are poised to trigger M phase. Once these complexes receive an additional signal, they become active and set the events of M phase in motion. The MPF complexes add phosphate tags to several different proteins in the nuclear envelope, resulting in its breakdown, and also activate targets that promote chromosome condensation and other M phase events. Here you can see that the CDK is getting activated when the M cyclin is attached to it, and the targets are phosphorylated, and then the M phase will begin with spindle formation, chromosome condensation, and the nuclear membrane breaks down. The anaphase promoting complex is cyclosome. In addition to driving the events of M phase, MPF also triggers its own destruction by activating the anaphase promoting complex or cyclosome. It is a protein complex that causes M cyclins to be destroyed starting in anaphase. The destruction of M cyclin pushes the cell out of mitosis, allowing the new daughter cells to enter G1. The APC also causes destruction of the proteins that hold the sister chromatids together, allowing them to separate in anaphase and move to opposite poles of the cell. APC is also an enzyme, but it has a different type of function than a CDK. Rather than attaching a phosphate group to its target, it adds a small protein called ubiquitin, that's UB. When a target is tagged with ubiquitin, it is sent to the proteasome, which can be thought of as the recycle bin of the cell, and destroyed. For example, the APC attaches a ubiquitin tag to M cyclins, causing them to be chopped up by the proteasome and allowing the newly forming daughter cells to enter G1 phase. Besides ubiquitin tagging, it is also used to trigger the separation of sister chromatids during mitosis. If the APC gets the right signals at metaphase, it starts off a chain of events that destroys cohesin, the protein glue that holds the sister chromatids together. The APC first adds a ubiquitin tag to a protein called securin, sending it for recycling. Securin normally binds to and inactivates a protein called separase. When securin is sent for recycling, separase becomes active and can do its job. Separase chops off the cohesin that holds sister chromatids together, allowing them to separate. The APC first adds a ubiquitin tag to a protein called securin, sending it for recycling in the proteasome, and the securin normally binds to and inactivates a protein called separase. When securin is sent for recycling, separase becomes active and can do its job. The separase chops up the cohesin that holds the sister chromatids together, allowing them to separate.